So it's a great pleasure to have Scooter Morris. So Scooter has been here for a couple of weeks. He's already been here for a week and a half and has been talking to lots of people and running a couple of workshops, and will be here until the end of this week. And so Scooter is the Executive Director of the Resource for Biocomputing, Visualization and Informatics, and also Adjunct Assistant Professor at the Department of Pharmaceutical Chemistry at the University of California, San Francisco.
Very good.
But also he's on the team for both Cytoscape and Chimera, and these were the subject of the two workshops that we've already been having. And today he's going to tell us about life is all about connections. So thank you very much.
Thank you. Thanks all for coming. I will start off just by thanking Elaine for taking care of me for the past week and a half and getting me situated, and also Matt for getting me here. Speaking of that, this is the outline. By the way, I should mention the slides are online. So I always put slides online. They will stay online. You don't have to download them or anything like that. If there's a slide you want to borrow because you want to make a point, grab it. It's yours. It's all on GitHub. You can just check it out if you want. I want to start talking about connections. And the first thing I want to do is get all the way up and think about how we connect. And I thought a fun way to do that is just to answer the question, how did I get here? And no, this is not about airlines. I'm going to go back into a little bit of history. A very long time ago, and you'll see the dates come up, I had a friend named David Shoemake, and he and I were in grad school together. And he was also a friend outside of grad school, and we did some interesting work together. And he got involved in a conference, this Chi conference, which was a little tiny conference at the time. And this was in 1985. And he asked me if I would be the AV chair for Chi 85. And I said, sure. Now, time happened. As a result of me being the AV chair for Chi 85, I got involved in this organization called SIG-Chi. And I thought it was very useful. I really liked some of the things that were happening in HCI at the time, even though it was sort of peripheral to what I was working on. Chi was sponsoring all of these various conferences year after year. And I was sort of got involved and helped with some of the conferences as a volunteer. And somewhere along the line, Matt decides to get involved in HCI, joins SIG-Chi, starts attending SIG-Chi conferences, and Matt and I sort of met each other until Chi 2014. Now at Chi 2014, Matt winds up being the co-chair. And, well, I was still the AV chair, but some of us moved on, some of us didn't, and Matt and I started working together. So, connections. The thing that we're going to talk about is connections. And I wanted to start off with connections between us, between colleagues, between collaborators, between organizations, because that's where we all start with connections. But this talk is mostly about connections in biology. So I want to go all the way down to a very low level. And then we're going to work our way back up. We won't get all the way up to the level that we just were talking about. But anybody know what this is? Okay. Everybody take a deep breath. You just used this. There we go. Hemoglobin. Now, this is the heme group right here. Right? You can see that heme group sitting there. And it is what we all use to carry oxygen. Great. What does this have to do with connections? It is a network. So we can take a look at this structure, render this structure as a network, and begin thinking about this in terms of graphs, in terms of graph theory, in terms of connections. Just to sort of give you a little bit of, there's the heme group right there. I changed the size and I added a little heme icon on there. Actually, that's the 2D structure of the heme group. And these are all of the residues. And the various colors on there are showing you the covalent bonds and the hydrogen bonds and how this network goes together. So at the very lowest layer of the proteins and the lipids in your body, we have a network, right? So that's where it's going to start. It all starts at that lowest layer network. And there's a zoom in on the heme group. But then it moves up. So now what we're going to do is take a look at a different structure. Okay, does anybody know what this structure is? We'll give you all just a shot.
Looks like cancer.
It looks like cancer. Good guess. Hopefully not.
Right, okay.
This is RNA polymerase. Okay, so RNA polymerase is the machine in your body, and what that machine does is it's going to read the DNA and make the RNA that's going to make the protein. Okay? So we've now gone from a very simple network structure, and now we're going to move up the scale to a molecular machine that is a key part of life. It's what's keeping you alive. And I'm going to take a simple, a little more abstract representation. These are the main components, the A chain and the B chain, and you can see how everything is connected. All of these dashed lines and solid lines are showing you the connectivity between these chains. And when I talk about chains, the best way to think of this is individual pieces of the protein that are independent and held together some way. Okay? We can talk about how that happens with hydrogen bonds and hydrophilic and hydrophobic regions and all sorts of things. But for our purposes, that's irrelevant. The key thing is we have another network. And we can go up from that. So now what you're looking at is these are all of the components. You can look at these as all of the chains, as it were, in this structure. And these are all of the interactions, other proteins that these chains interact with. So I'm zooming one step out. And at each step, we're getting more complexity and we're getting more functionality. So until we get to this. So you were interested in a large network. So that's the human interactome. So this is all of the known interactions trimmed at a 0.8 level, cut down. And what I've done to sort of highlight some things is those proteins that we were looking at before, I made them much larger and I colored them. And there they are right there. Okay? And this gives you an idea of the complexity when we're starting to talk about how all of us work. What are the connections that are going on there? How do these interact with everything else in your body in order to generate the machine that is you? Okay. So, anybody have a guess of what connections I'm looking at here? Brain. There we go. So up until this point, we've been really talking about how proteins interact. So the previous image was a protein-protein interaction network. And now I've come up a level still, and now I'm talking about a cell-cell interaction network. We're talking about how the neurons, the central neurons in your brain, sort of are connected. And we could zoom out, we could add a lot more detail. Obviously, it's much, much more complicated than this. This happens to be a nice image that I was able to find that still looked like a brain. And we can start thinking about cell-cell interactions. And again, at each step, I'm sort of taking a step back and zooming out. How do cells interact? I think I've sort of given you a clue on how proteins interact. There are these cell gap junctions when cells are actually together. There are tight junctions when cells are together but they're passing information between them. And then there's these desmosomes. All of these are three ways that cells interact with each other when they're together. But the big part, and the part that I think is really intriguing, is how cells interact at distance. So everybody knows about hormones, right? So the hormones are actually ways that part of your body is communicating with the rest of your body. And there are these signaling mechanisms. This is a pathway. We're going to talk a little bit more about pathways. And you can see all of these are various factors that are coming in and telling this cell to do something. Giving this cell a clue about environmental factors, about, hey, you should die, please. And that's part of a very critical mechanism, the apoptotic mechanism, where I need to tell this cell that what it's doing right now is not all right and I want it to go kill itself. Okay? And one of the things that has to happen in cancer is cancer has to turn that mechanism off. So it's really important to understand how this cell-cell communication works. Okay, why do we care? If we're thinking about all of these kinds of interactions, why do we care about these connections? And here's a great quote. Empirically derived networks are necessary for describing the molecular mechanisms and biological processes that drive disease. So one of the reasons we care is because we need to do it to understand disease, right? So that's a useful thing. Let's think about this network. This network was published initially as part of the Cancer Genome Atlas, TCGA, and it's looking at all of the key mutated genes in glioblastoma. Now, if I want to treat glioblastoma, right, I'm going to have to treat what's going wrong in glioblastoma. So, which single gene is responsible for glioblastoma? There isn't one. The message here, and if you look at all of these are all mutated. Every single one of these is mutated. So we've highlighted the ones that are mutated the most, but the point here is that glioblastoma is recognized as a network disease. These cancers don't have to just mutate one single gene. They can mutate something in a pathway, and they can disrupt that pathway, and once they've disrupted the pathway, then they can progress into other cancers, into all of the downstream things that are happening. So this is why it's really, one of the reasons it's absolutely critical that we understand interactions and connections between proteins and between cells. Okay, why else do we care? Careers. At the end of the day, citations. I counted, not one at a time, but I did a PubMed query. And in the last decade, there's been 83,000 papers that with the mesh term, so this is not even actually going down and looking for abstracts, that specifically have the mesh term related to pathways or networks. And I explicitly excluded, just so that we're all clear, all of the machine learning, deep learning, neural network stuff. I pulled all of that out. So 83,000 papers, right? So some of those, and I'll mention a couple when we go on, there's a lot of opportunity for career advancement in this area. Okay. So in the abstract of the talk, that I first did, I made sure to talk about the fact that I was going to spend time on all of how you go in and find these connections and all of the current technology for doing that, and get into depths on this. And I realized that I really didn't have a 2-hour window to do this talk. So what I decided to do instead is I'm going to just sort of mention some of these techniques. The links on this site go off onto detailed series of slides that explain each of the technologies in detail. Okay, so I will just encourage you, if you're interested in some of this stuff, feel free to head off and go on these links and go down to them. But what I want to start with is, how do we really know how things interact, what the connections are? And the gold standard here that everybody will talk about and they'll mention is X-ray crystallography. So essentially what you do in X-ray crystallography is you're going to make a crystal. And when we're talking about interactions, you're going to make a co-crystal. So somehow you're going to get these two things, whatever you're trying to determine, connect, to crystallize together. And once you've done that, you can actually get a atomic resolution map that's like hemoglobin that we saw before, that will tell you how these things fit. All right, so that is sort of the gold standard. Now, just so that everybody understands the problem with gold standards, like this, is X-ray crystallography, in order to get things to crystallize, all right, often what you have to do is you really have to change the structure. So X-ray crystallographers, what they'll do is there'll be little floppy bits that'll, are very flexible. Well, they have to cut those off. And sometimes to get it crystallized, they'll introduce a lot of ions and other kinds of molecules, because the challenge here is proteins are flexible. And in order to get it to crystallize, you can't have it flexible. So, but nevertheless, for getting an atomic resolution structure, that's the way to do it. I want to talk just a bit about, these are all other techniques. Computational docking is sort of a big area. These days, everybody's talking about cryo-EM. And the reason that they're talking about cryo-EM is in the last 5 to 10 years, there's been a huge improvement in the resolution of cryo-EM structures. Cryo-EM is different than X-ray crystallography in that what you'll do is you'll take a large complex or structure that's together and you freeze it. So rather than trying to crystallize it, now what we're going to do is we're going to freeze it. And then we put it in a machine, and we do, so we're going to do basically micrographs. We're going to make images of that. And as we make the images, we're going to move it. So we can get the images from a number of different angles. And there's many of the structure generally in the plate. So we're going to average all that together, and we're going to do refinement of that. And we get now, we've gone from 10 angstrom resolution down to 3 and 2 angstrom resolution. So X-ray is about 1.5, 1.8 angstrom. So now we're starting to get cryo-EM where we don't have to lop bits off. We don't have to make huge modifications to the structure. We're actually able now with cryo-EM to get down to an area where we're really able to get some images of these large machines. The other problem with X-ray crystallography is we're limited by size. Okay, so cryo-EM, some of the big structures structure that I showed you of RNA polymerase was done with cryo-EM. And the proteasome, which is even a larger structure, has now been done with cryo-EM. Okay, so that's looking at the structural level. Do these two things go together? Now, when I say this, I'm sort of looking at and talking about how specifically they go together, but knowing that two things bind, right? There's a whole bunch of binding assays. And I'm certainly not here to say that traditional biochemistry is dead, and that traditional binding assays and all of the things that have led us up to here are passé because they're still critical and they're still used quite heavily. But high throughput techniques like yeast-2 hybrid and APMS have really revolutionized some of the ways that we look at this. So yeast-2 hybrid is a very simple mechanism, and we can run a lot of yeast-2 hybrid experiments. Basically, you're taking a bait, and I won't go into the details, but you just let things bind, and then you have a readout that will tell you whether or not that these two things are together. And you can do that, basically for lots and lots of things. I can keep using different baits, and then eventually I get a network out. APMS, affinity purification mass spectrometry, is a very similar kinds of approach, except now instead of using a yeast vector specifically, I'm sort of doing high-level mass spectrometry. I'm just binding them, and then I'm just going through the mass spec and figuring out if these things are together. All right. So I'm not going to talk about single-cell genomics. I may mention this later, but cell-cell interactions, there's just, wow, there are all sorts of different ways. Imaging techniques that are coming, various kinds of secretory assays where we're figuring out that this particular cell is secreting, and then we can find out later on that this particular cell has a receptor for that. Microfluidic approaches, I want to get back to that, but it's critical to recognize how much of all of this stuff has benefited from bioengineering. So the microfluidics that has come out recently from the engineers has really helped us in terms of the kinds of experiments that we can run. And then host pathogen interaction with host pathogen proteomics is where we're getting interspecies, and that's huge. That's how, and I think I have a slide a little bit later to talk about that. Okay, so I know that it's really interesting to work on problems that have big data. And because you can, big data is the thing. Everybody's talking about it. You can use machine learning algorithms. You can do all of this stuff. So I want to talk a little bit about really big data, just so that we're clear. So the number of proteins in a human cell is 1 × 10⁻⁻⁻. The number of lipids in a human cell is about 1 × 10⁻⁻⁻. The number of metabolites is 1 × 10⁸. The number of RNA molecules is about 1 × 10⁸. The number of miRNA is about 1 × 10⁴. So as a back of the envelope calculation, we're dealing with 1 × 10⁻40 molecular entities in every cell in your body. The number of cells in a human body is roughly 3.7 × 10⁻13. Okay, 1.3 × 10⁻107. That's a big number, just for the record. You want to deal with big data? There you go. Now, the reality is that we are nowhere near that amount of interactions. And the reason is that our cells are heavily compartmentalized. So the proteins within our cell are all divided up. And then our cells, not all the proteins that are on our cells communicate with other cells. And then we're divided up into tissues. And the tissues, there's tissue specificity there. So basically, it's much smaller than this, but still it gives you an idea on the scope of the data sets that we get to deal with. But wait, there's more. Connections aren't static, right? So these connections all vary on time, development, age, unfortunately, time of day, the physiological changes that you might have. environmental conditions, and disease. Organisms connect also. We talked a little bit about that. And here's the image that I mentioned before. So this is an APMS study looking at HIV infection. So what they've done here is they took a culture of human cells, and then they intentionally infected it with all of the HIV proteins. And then out of that, they could say which HIV proteins are interacting with human proteins. So everybody knows, right, HIV is a virus. It does not have a way to reproduce itself. So it has to hijack your cells in order to do that. And this is what this is trying to get at, is how does it hijack your cells? So one of the outcomes of this study in particular is VIF right here. It wasn't known that VIF interacted with COL5 and what that mechanism was. So that was a new result that came out of this study, and it's led to a lot of further research on VIF in particular. These kinds of networks, these kinds of connections, can help us understand how to solve really serious human diseases. So that's one of the reasons we really care about this stuff. Okay. So, how do we deal with complexity? So how do biologists do this? How do we help them do this? So, first thing that everybody should know is we're good, we, meaning biologists, I'm going to wear my biologist hat, are good at simplifying things. We have to. We cannot deal with this. And one of the first ways that we do this is abstraction. We take these complexities and we start thinking very seriously on how we can pull out key things that we want to say about that complexity, and then we draw these diagrams. So here is, can't even remember which one I pulled for this. It's a metabolic pathway, but I can't remember which one right now. But here's the glucogenesis. Here is the TCA cycle. We take these and we do abstractions. Now, we do this as humans all the time. And my favorite example, I should have put in an image. Rats. My favorite example of that is how many of you have, it's a stupid question, but okay, we're going to wake everybody up. How many of you have been to London? Great. How many of you have used the tube? Great. How many of you have used the tube map? Great. How many of you have used the tube map to navigate around London without using the tube? Really? I'm impressed. Because the Thames does not go like that. So what's going on? Why is the tube map so useful? Why does everybody talk about it? Why, when they put up that map, everybody knows right away that it's the London Underground? What makes it useful? It is not a map. It is a diagram. It's an abstraction, and it's an intentional abstraction, and that's what these pathways are. It's a way that we can abstract things out and highlight what we're trying to talk about without the fact that there are, let's see, where is it? IDH isn't even in this particular one. IDH2 is one of the proteins that are involved in the TCA cycle, IDH2 and IDH1. And it turns out that those two are also involved heavily in glioblastoma. Well, we're not going to build all of that information into here because it's not relevant to this abstraction. So the first way that we're able to deal with some of this complexity is we abstract it out. We're going to leave it. Thank you. The other thing that we do, and hopefully this is where now the computer scientists and the mathematicians are going to wake up. It's time, your time, guys and ladies. Here is a network. Is there a topology? Is there a structure there? Don't know. I don't know either. Well, yes, I do, because I have done the clustering to find it. So what we'll do is we'll use clustering algorithms, and in these cases, these are unsupervised, although a lot of people are getting interested in supervised methods, to find tight groups and to find the topology that's hidden with that, in that, all of that complexity. And then we'll pick a part of that that might be of interest, and we can analyze each of these separately. So in this case, what's going on is that is the same cluster that I showed before, except now we're doing enrichment analysis to find out which terms are enriched in this cluster. And what you can see is that the mRNA signal processing and spliceosome are the two terms that are highly enriched here. And we can begin making biological inference about that. Same thing. Just so that when we talk about a network, all the Viz people know this, but I'll just make it clear. When we talk about a network, we don't necessarily mean a node link diagram. This is a matrix representation, but essentially what you're looking at is nodes across the top and the side. And if you see color, then there's a connection between them. So in here are the edges. And in this particular representation, you can clearly see the topology. So this is a great alternative way to look at some of this data. That was topology driven, but we can also use attribute driven. So this is a study that, it's a network that I put together. This is looking at, I think, 276 breast cancer patients. And so we have, we have patients along here, and we have genes down here. And we're looking at expression profiles across here. And this is just a standard k-means clustering of that. And what you can see is those patients are falling into 4 different categories. And breast cancer does tend to break up into 4 different categories. And we can use this. Then once we have this attribute-based clustering, we can use other techniques and link it back to the pathway or the network. And of course, we can do the hybrid. And I think that this is an area that there's just a lot of opportunity for work. So what's going on in this particular network is there are 2 types of clustering going on. One where you can see the nodes have some sort of attribute painted on them. But on top of that, we're looking at the topology. So we're combining both an attribute-based metric and the topology to find modules or subnetworks of interest. By the way, when I say modules, I mean communities. When I say networks, I mean graphs. It's just, unfortunately, we all use different terminology, but that's the terminology. And then, of course, more recently, machine learning techniques. I think this is an interesting one where what they're doing is they've got a number of different features that they're piping in, and they're getting patient similarity networks based upon those features. And then they combine those, and you get an integrated patient similarity network, and then you can stratify. And the nice thing about this is that they're able to pull from this. This is something called NetDX. I've given the link if people are interested in looking at that. Is the way that this works is it's not black box. Because I have those patient similarity networks, I can work my way back, and I can figure out why the stratification came out the way it did. Okay, and for the HCI people, interaction is the other way that we really deal with some of this complexity. And all of your various favorite ways of doing interaction, so overview plus detail, focus plus context, the thing that I use heavily and sort of encourage is a combination of linked views and brushing and linking. So I'll bring up a heat map, which is showing perhaps an attribute-based clustering, a hierarchical cluster based upon attributes. And that will be linked to a protein-protein interaction network. And now I can go back and forth and explore those. Or I'll bring up a structure like the structure that we saw to begin with of hemoglobin. And I'll bring up the network of hemoglobin and the same thing. Those two are linked displays, and I can sort of explore them and take a look at the network dynamics and also take a look at the structural dynamics. So, all right. Let's see, how am I doing on time? Okay, good. So what I wanted to do is talk a little bit about the stuff that we're doing right now, and then the challenge at the end. So, first thing, some of you have seen us playing with Chimera, Chimera X. You can see that what we're working on here and what we're really trying to do is take these structures and provide some of the connections, some of the tools that you can pull out connections from the way that these things pull together. Here's an even better example where what we're looking at, see, if you see these little green lines, they represent chemical crosslinks, because that's one of the ways that we can come up with how things are connected using chemical crosslinking when we don't have solved structures. The other tool is Cytoscape. Cytoscape allows us to see the graphs and to do calculations on the graphs and to bring known protein-protein interaction data in. Some of you were in the Cytoscape workshop. You've seen this. You've played with it. So I won't go into too much detail. But what I do want to talk about is, oh, and this is what I mentioned before. This is something called Cluster Maker. Two things that I forgot to mention. For those of you who are interested, for the computer science folks in particular, and you sort of wonder about building tools and having to support those tools and how much effort that is. So Cytoscape has 20,000 citations. Chimera has 20,000 citations. If citations are important, and I think for all of us in academia they are, you can get a lot of citations with this effort, but you can't do it alone. You need the collaboration. This is something called Cluster Maker, and Cluster Maker does not have 20,000 citations, unfortunately. It only has 250 citations, but this is a tool that I wrote to layer on top of Cytoscape to give you this linking. So this is 3 different types of data sets. Here you have a clustered protein-protein interaction network. Here you have a genetic interaction network. And here you have a heat map that's showing an attribute data set. OK. Now I'm going to talk about some new work very briefly. There's something called SCNetViz. How many of you are familiar with single-cell genomics, single-cell RNA-seq? Clean sweep. OK. Very quickly, imagine for a moment, everybody knows about genomics and getting your sequence and going to 23andMe or pick your favorite. All right. So imagine for a moment that it was possible that I could tell you every cell, down to the cellular level, what genes are being transcribed. OK? So that's what single-cell RNA-seq is. So the problem is that I have all of this data, and for example, let's say I have 100,000 cells that I'm getting data, 27,000 genes. That's a big matrix. How can I analyze this? How can I look at this? using some of the techniques that we've talked about. So basically what we do is we take the matrix, this is this massive matrix, and we cluster it, and then we do and go and find protein-protein interactions on the clusters. And you can go through and look at each one of these, do the network analysis on it, do all of the enrichment analysis on it, and of course you can do all of the kinds of plots that you'd like to do, and all of these things are linked. And I can go back and forth between this data and really explore these huge data sets, but with an abstraction that sort of makes sense. Spoke, the last big thing that I want to mention, because it's sort of taking networks and connections to the next extreme. There's lots of public databases out there. The problem is they're not linked. So how do you navigate between protein-protein interaction data and disease data? Or how do I navigate between expression data and proteins and disease? And the idea is to build a system, a massive, massive network. That's what this is. And the goal is a big knowledge network that combines all of this data. This gives you an example of a query. I've queried for this protein, and I get out this network. And now I can find, here's the protein right there. This is, I'm hovering over this to get an edge type. I'm showing what it binds to. I'm showing the diseases, and I can begin exploring this network and working my way out. And this is, I took out one more step just to show you how all of this gets explored. And this gives you an idea on how the data that we're pulling in. Each one of these types has different data sources. So we're doing, this is just a massive bioinformatics, pull the data in, try and link up all of the various identifiers so that they make sense, and put it into the network. The big things that we're working on right now are differential and dynamic networks. Those are the hard problems. There's some, our new grant for Cytoscape has just gone in. There's a lot of discussion about how we're going to approach these two things. Okay, wrapping up. So, understanding biological connections, hopefully everybody's got it. We should do it. It's a good thing to do. It's critical. Great. I won't belabor that point. What's it going to take? It's going to take new computational algorithms. It's going to take new math. It's going to take new approaches to visualization. It's going to take collaboration. So collaboration is the only way we're going to solve these problems. I will tell you that I am not a mathematician. I am mathematically challenged. I need help when it comes to really thinking through some of the algorithms that I need to do and work with. And having collaborators who are knowledgeable in mathematics would be just critical. So single-cell RNA-seq, this brand new technology that everybody is really excited about right now, took collaboration between engineering to do the microfluidics. It took collaboration on biology to understand the genomics. It took collaboration on math to be able to analyze this data, because the data is really sparse and really noisy. And a lot of collaboration with computer science. So all of that had to come together in order to get us this new technology. All right. So this is a week and a half of observation. Okay, so, you know, if I offend anybody, just take that with a grain of salt. But I've been here for a week and a half, and so here are some observations. Swansea University has some amazing biology going on. I was on the other campus for a day and have 2 collaborations. So I'm pretty excited about it. And that includes some really intriguing ecological succession work going on. that has to do with how species compete and outcompete each other on the one hand, and on the other hand, looking specifically at cancer and a potential therapeutic for cancer, and having to do some molecular docking on that. So that was 2 meetings. So there's some really intriguing work going on there. A lot of interesting medical sciences. Meeting and talking with some of you here on this campus. There's a lot of really exciting mathematics and computer science going on. But it doesn't have a program that brings everybody together. And I think, given what you've got, it's really surprising to me. I come from an organization, UCSF only has medical. There are no undergraduates. And sort of computational biology is sort of in our, I won't say in our DNA, that's just, no. But it's really just sort of inculcated to the extent that the university is underwriting the development of a 20,000 core cluster to be used by everybody, that any user can sign up without paying. Some ideas. How do we do, what's next? Start small. How about a computational biology colloquium series? The biomass series, which this is, right? I'm stretching math really far here. Thank you, Elaine. But that would, in collaboration with the biomass series, let's get the computer scientists. Let's really start tackling some of these in collaboration with some of the biologists. Some of the challenges with molecular docking would be a really intriguing place to start. Lots of weird geometry stuff there, and how all that fits together. More cross-campus events. How could, maybe courses that are over there, courses that are over here, 1-day workshops or 3-day workshops or something to get things cross. And this one's a little a stretch. Maybe an open door policy to encourage collaboration. So I don't know whether it's just the time of year, term end, whatever. Interesting point. I figured that out too, and I went and dug up a doorstop. And what I found, so first couple days, right? Elaine found an office for me, which was fantastic. Thank you very much. I was able to get a lot of stuff done, but the door kept closing. And so when people wanted to come give me a badge or something like that, what would they do? Knock on the door, right? So I'm in the middle of doing something, and so now I get up, I open the door, and do they come in? Do I, at any rate, door stops. I would heavily invest in door stops. That's kind of my take. So, however, I will point out that overall, you guys get networks. You understand connections are really, really important. I would just like to see some, maybe some deep collaborations going down into biology somehow. And with that, I'll thank everybody for their attention, and we can have a discussion about all of that.